Good morning, all. Glad to see you here again. Uh, let's get started with the lecture. It's 9:30. Do you have any questions, comments before I get to the reminders in the material? Okay, great. Moving on. All right, so reminders. Quiz 3 and is available for you to take anytime between now and Friday, 11:59 p.m. Quiz 4 opens immediately after. It opens midnight on Friday, so technically it's going to be Saturday. And you can take it anytime between that, so between Saturday and Monday, February 22nd. So you will have a bit more time to take with the Quiz 4. And the additional assignment is due today, but I think I'm going to extend the deadline until Friday too, because a lot of people have emailed me that they have some sort of issues with lockdown browser, and some of them are related to Mac systems, some of them are related to having a Chromebook. So what I recommend you do if you are one of those people is please contact Web Courses support. You can either chat with them online or email them or call them, but please figure it out before Monday. So I'm not a technical person. I cannot necessarily help you out figure out what the problem is, but UCF support is. But it is important for me to know if you're still having issues. So if you're still having issues, please tell me what the issues are and how did Web Courses support comment on it? How did Web Courses support suggest you solve the issue? All right, so that's all for the reminders. Today's lecture is going to be on meiosis. We've covered binary fission. We've covered two cell division types so far. The first one is binary fission. The second one is mitosis. And now it's time to cover the third one, meiosis. All right. So life cycle includes both life cycle of eukaryotes, or organisms that have cells with the true nucleus. It includes both mitosis and meiosis. So take a look at this cell here. This is let's say this is a zygote. It's a fertilized cell, or the origin cell from which our bodies develop. So this zygote, original cell, is going to divide, and the cells are going to specialize eventually into and will eventually look like an embryo, and eventually look like a human being. But in the very beginning, the cells divide mitotically. Okay, so the mitosis is going on during the embryo development. Once the embryo is a human baby and the human baby grows up, another type of division is going on, apart from mitosis. Another type of division is going on in the ovaries or testes of a human being from the germ cells. And ovaries and testes contain so-called germ cells, which divide meiotically. Okay, so the meiosis is going on only in the germ cells, which are located in ovaries and testes. And during meiosis, the diploid cell produces 4 haploid cells. So one diploid germ cell produces 4 haploid sex cells that are also called gametes. So meiosis is needed for gamete formation. And after gametes are formed, and if female gamete meets the male gamete, fertilization occurs and the zygote forms, and the process repeats all over again. All right, so meiosis is somewhat similar to mitosis, except for it's not producing genetically identical cells, and it's producing haploid cells instead of the diploid cells, unlike mitosis. So zygotes, which zygotes is a fertilized egg. It's the cell that fuses male and female gametes. And zygotes are diploid, and they have 2 full sets of chromosomes, one from each gamete. So this is a reminder what diploid means Remember, in humans, we have 23 pairs of chromosomes. One chromosome out of the pair is inherited from the mother and another inherited from the father. So if if a cell has both members of the pair, the cell is considered diploid. If the cell has only one member of the pair of chromosomes, it is considered to be, it is called haploid. So the zygote will undergo mitosis, not meiosis, as I just mentioned in the previous slide, to develop into an adult. So here's the origin. After fertilization, the zygote is formed, and the zygote is a diploid cell, the origin cell, right? The zygote starts dividing mitotically. Eventually, the cells that were the products of mitosis are going to specialize, forming an embryo, then a baby. Once the baby is formed, the cells are going to divide even more mitotically, and once the individual is sexually mature, the production of gamete begins. So the gametes are produced from the germ cells in the testes or ovaries, and the cycle repeats again. So haploid gametes give offspring the right chromosome number. If the gametes were diploid, then the zygote would not be diploid. It would have more. It would not have pairs of chromosomes. It would have like chromosomes in threes or in fours, depending. Like if one gamete is diploid and another gamete is haploid, we would have the zygote that would be noted as 3N, or it would have not a pair of chromosomes, but a triplet of chromosome. So that does not happen, at least that does not happen very often. There are some genetic diseases there are some diseases that are caused by this, but by having not a pair of chromosomes in the zygote, but a triplet or a missing chromosome. But the point is, the haploid sex cells produce a diploid zygote. If the sex cells were not haploid, then the zygote would be messed up. Then there would be some serious issues with the development of the zygote, because there would be not a right number of chromosomes in it. So in humans, the sperm cells are carrying 23 chromosomes, not 23 pairs, but only 23 chromosomes. They fuse with the egg cells, which also contain 23 chromosomes, not 23 pairs of chromosomes. And in result, these two zygotes give, gametes give a zygote that has 46 chromosomes, or 23 pairs of chromosomes. We have 23 homologous pairs of chromosomes. All right, I see a lot of messages in the chat, and Ansley is covering it, and that is okay. Any questions so far? All right. Meiosis is similar to mitosis, but there are some differences still. So how is it similar to mitosis? Well, first, both are forms of cell division with similar phases, but in meiosis, these phases are repeated twice. So interphase occurs before the cell divides, and it is similar in meiosis and mitosis. During the division, the cell goes through prophase, metaphase, anaphase, and telophase. So it's the same in meiosis, except for there's two phases for prophase. There's two stages for prophase, two stages for metaphase, two stages for anaphase, and two stages for telophase. As you will see in the following slide, there's a meiosis 1 and meiosis, well, here it says that there's meiosis 1 and meiosis 2. So in meiosis 1, prophase is noted as prophase 1, metaphase is noted as metaphase 1, anaphase 1, and telophase 1. And in meiosis 2, there is correspondingly prophase 2, metaphase 2, anaphase 2, and telophase 2. And finally, the final way that meiosis is similar to mitosis is through cytokinesis. And cytokinesis, a reminder, is a splitting of one cell into two daughter cells. So cytokinesis takes place at the end of each cell division. So meiosis also well, I covered that. Is how it's different a bit. Let's take a look at what's going on in more detail. So in meiosis, DNA replicates once, but the nucleus divides twice. In mitosis, DNA replicates once and the nucleus divides once. Okay? In meiosis, because DNA replicates once and the nucleus divides twice, we're getting 4 haploid cell out of 1 diploid cell. So similar to mitosis, DNA replicates during an S phase or synthesis phase of the cell existence. And it occurs before the cells start to divide. Remember, interphase. The S phase is located within an interphase of the cell cycle, and it's located in between the 2 gap phases. So during gap 1 phase, a reminder, the daughter cell has just started existing, and it performs its regular functions, whatever, depending on where the cell is located. During the S phase, the DNA is replicated, and during gap 2 phase, or G2 phase, the cell grows some more and synthesize necessary chemicals to undergo cell division, mitosis or meiosis. So let's see. This is a high level overview of what is going on during meiosis. So during meiosis, during prophase, we have the spindles are forming, the DNA, the DNA condenses around the chromosomes, and during metaphase, the chromosome and the nucleus nuclear membrane dissolves. During the anaphase, the chromosomes line up along the equator of the cell, and here it jumps straight to cytokinesis. I guess there's a stage missing here. Anyway, I will go over the stages in the slides in more detail. So similar to mitosis, to the prophase in mitosis, in prophase 1, the spindle forms, the chromosomes condense and attach to it. So there's also early prophase 1 and late prophase 1. During early prophase 1, we have the chromosomes condensing and becoming visible. And in the late prophase, the nuclear envelope breaks up, or the nuclear membrane breaks up. The crossing over occurs and the spindle forms. Now, crossing over I have not gone across, but it is pretty fascinating stuff. And crossing over is a process of mixing, of exchanging of the genes between the neighboring chromosomes. And it is one of the ways how genetic diversity of the offspring is kind of guaranteed, is produced. Okay, so in prophase 1, homologous chromosomes also pair up, and it happens only in meiosis. Remember, in mitosis, the chromosomes did not need to kind of pair up. They were just replicated and pulled apart into different cells. But during meiosis, they pair up. Each chromosome lines up next to its homolog. Well, the homologous chromosomes line up. Now, crossing over that occurs in meiosis prophase 1 is depicted in this slide. The homologous chromosomes pair up precisely, aligning gene by gene. And during crossing over, pieces of homologous chromosomes physically change places. So here are 2 homologous chromosomes pairing up. The blue one is inherited from the father, the red one is inherited from the mother. And these, the chromosomes can literally cross over, hence the name of the process, and exchange the alleles of the genes that are located in that particular locus. So as a result, we still have 2 homologous chromosomes, but they are not uniquely inherited from the mother or the father anymore. Note the change of color in the X chromosome here, right? So most of the chromosome originally inherited from the father conserve the genes that were inherited from the father, but it also has some alleles of the genes that were inherited from the mother. And the chromosome that was originally inherited from the mother now contains, after crossing over, it contains the genes that, alleles of the genes that were inherited from the father. So as a result, the gametes that have been formed from the chromosome originally inherited from the father will contain some genes, some alleles of the genes that were inherited from the mother. And so this is like not 100%. If you inherited one chromosome from the father and one from the mother, you may not necessarily produce gametes that were uniquely, that contain uniquely the genes from the father or uniquely the genes from the mother. Because of this crossing over process, the genes mix, the alleles for the genes mix up, and producing some new combinations of alleles that would be unique to you, unique to your gametes that you produce, and not the gametes that your mother or father produced. All right. So, and this process, this crossing over again, it's basically shuffles the gene and contributes to genetic variability. It contributes to increasing genetic diversity, and increase so by producing new combinations of genes and associated traits. I suspect this crossing over process is familiar to you already from grade school, but if you have any questions, please do not hesitate to ask them now. Okay, you guys have gone quiet for the past couple of days. It's I take it either you know about this and you're just sitting there, just move on already, or I'm being too I'm explaining it not well enough. All right, so moving on. So metaphase, in metaphase one, in meiosis, chromosomes line up in two rows. So it's similar with metaphase in mitosis, except for in mitosis, the chromosomes line up in the single row, whereas in meiosis, the chromosomes first pair up to go into homologous pairs, and then they line up across the equator. So note that the spindle fibers spindle fibers are responsible for this lining up of chromosomes along the equator. So the spindle fibers during prophase, once the nuclear envelope or nuclear membrane is dissolved and chromosomes kind of swim around in the cytoplasm, spindle fibers grab onto them during the late prophase. And during the metaphase, the spindle fibers line the homologous chromosomes along the equator. So in this metaphase one, there is one fun fact that I want to mention to you at this point. We have made, in agriculture in particularly, we have made a lot of hybrids of plants and animals. And not all of the hybrids that we've produced are fertile. A very common example of this infertile hybrid that was produced by humans is the mule. And a mule is a cross between a horse and a donkey. It's a working animal, very useful, but sterile. The two mules the two mules can never produce a mule, so they cannot become they're not fertile. Okay, the only way to produce another mule is to cross a donkey and a horse again. And why is that? The reason for that is that donkeys have 31 pairs of chromosomes and the horses have 32 pairs of chromosomes. So as a result, the mules have 63 chromosomes. So they have the odd number of chromosomes, and the chromosomes cannot pair up. So there will be always one extra chromosome. So the chromosomes cannot pair up during the prophase one, and they cannot kind of line up properly during metaphase one. And because of this, they cannot produce gametes. Like their gametes are just not functional. The meiosis breaks down, and if meiosis is not completed, then no sex cells are produced, and no reproduction can take place. So the mules, that's why the mules are infertile. It's because they have the odd number of chromosomes that cannot line up correctly and cannot be separated into haploid gametes. So a little fun fact to break it up from the dry explanation of the different phases of meiosis. So during metaphase 1, when the alignment of the chromosomes along the cell's equator occurs, the process called independent assortment takes place. And independent assortment is the second way the genetic diversity is generated. So the first way, remember, is the crossing over. Crossing over is the first way that meiosis that contributes to genetic diversity of the offspring, and this independent assortment is the second way, and it occurs during metaphase 1. During an independent assortment, chromosomes pairs align randomly, scrambling the combination of chromosomes in each gamete. So take a closer look at the picture here. So again, the chromosomes pair up into homologous pairs during meiosis, and these pairs can be arranged in various combinations in terms of where the paternal chromosome is located and where the maternal chromosome is located. For instance, the version 1, all of the paternal chromosomes are aligned in the top, and all of the maternal chromosomes align in the bottom. Version number 2, there will be on the top there will be paternal, and then maternal chromosome, and on the bottom there will be maternal, and then paternal chromosome. Version 3, on top can be paternal, and on the bottom there could be maternal, maternal, and so on. So there are 4 possible ways if you have 3 pairs. If your genotype contains the 3 pairs of chromosomes, there's 4 ways how the gametes can be formed. Okay, so note the end result of the gametes. So in the version 1, there are 2 possible gametes. In version 2, there are another 2 possibilities for gametes. In version 3, there's yet another possibilities for gametes, and so on. So possible easy way to calculate the possible versions of gametes is 2 to the power of 3, where 2 is the members of chromosomes in a pair. There are 2 members in the pair, and 3 indicates the number of pairs of chromosomes in a genotype of an organisms. So if the humans have 23 pairs of chromosomes, so to calculate the number of possible gametes in humans, or versions of gametes in humans, we would need to raise 2 to the power of 23. And that produces a fairly huge number. It's around 8.4 million. So just take a moment for that to sink in. Each individual organism, like you and I, can produce 8.4 million versions of a gamete. So this kind of explains how 2 parents can have 2 totally different looking babies, because their gametes can have 8.4 million variants of what kind of genes the baby will have. So it's pretty fascinating. So, and independent assortment is more important in creating genetic diversity compared to crossing over. Crossing over is kind of important, but is not going to produce as much genetic diversity as independent assortment. Okay, any questions about the independent assortment or crossing over, or anything I've covered so far, guys?
On our exam or anything like that, are we going to need to be able to do that calculation?
Not on this one, but in the following, on the final exam, there will be a question requiring understanding, like what the number means or why it was calculated. Well, I'm not going to ask you why it was calculated that way. Anyway, You will need to know what contributes to genetic diversity and how a particular number of chromosomes, how a particular number of gametes was generated.
Okay,
But
and then
not for this exam. Go ahead, Ansley.
I believe there's also a hand raised.
Oh, okay.
I don't know how to
Go,
pronounce your name.
go ahead. Oh, whoever raised their hand, I'll let me see. Anel?
Yeah, hi.
Hi. Go ahead.
I have a question. So, is crossing over and independent assortment, do they both occur, or is it like one or the other?
Oh, they both occur. So during meiosis, everything like both crossing over and independent assortment occurs.
So first it crosses over in prophase 1, and then independent assortment occurs after, correct?
That's right. So during prophase, crossing over occurs when the chromosomes line up together. First line up together, the crossing over occurs, and the chromosomes line up together during prophase 1. And during metaphase 1, once they're kind of they're aligned along the equator in a different way. So this alignment in the equator during metaphase 1, this is when independent assortment occurs. So independent assortment is basically the possible ways the chromosomes can be lined up along the equator.
Okay, thank you.
Yeah, no problem. Any other questions?
so both independent assortment and crossing over are ways that genetic diversity is achieved.
That's exactly right.
Okay.
Yes. And there is a third way, but I didn't get to it yet. So there are 3 ways the genetic diversity is generated. One way, the first way is crossing over. The second way is independent assortment of the chromosomes during metaphase 1. And the third way is random fertilization, but we're not there yet. Okay, so I'm gonna look through the chat for a minute. I see a question from Madeline. So will there always be 3 pairs of chromosomes in diploid cell, or is it different? It depends on the genotype of an organism. So this particular cell, this particular example, is of some hypothetical organism whose genotype consists of 3 pairs of chromosomes. In a human genome consists of 23 pairs of chromosomes, but it wouldn't be as easily to picture in a figure compared to an organism, a hypothetical organism with 3 pairs of chromosomes. So if we were to draw a cell of a human, okay, then we would need to picture not 3 pairs, but 23 pairs of chromosomes lining up during metaphase 1, and the number of possible variants for independent assortment would not be a neat number of 4. It would be a messy number of 8.4 million. Okay, so it depends on the chromosomes. The bottom line to your answer, Madeline, there will not always be 3 pairs of chromosomes in a diploid cell. There can be 23 pairs or 8 pairs or whatever the genotype is. Maybe it was confusing what a diploid cell means. The diploid term itself means that there is a pair of chromosomes in a cell, and one member of a pair was inherited from the mother, and another member of the pair was inherited from the father. So diploid refers just to the presence of the pair of chromosomes. If there is no pair of chromosomes, then the cell is haploid. So if there is only one member of a former pair of the chromosomes in a cell, then the cell is considered to be haploid. So this cell is diploid, but these resulting gametes are haploid because only one member of the former pair is present in the resulting gamete. Okay. And now I just covered the diploid, haploid, and I wonder if that cleared it up. I see your question in the chat here. Ansley's covered it. Great. All right, moving on. After metaphase 1 comes the anaphase 1, in which the cells are pulled apart by not the cells, pardon me, the chromosome pairs are pulled, the members of the pairs are pulled apart by the spindle fibers into the polar opposite sides of a cell, okay? And during the telophase is kind of reverse from the prophase. And during telophase 1, the nuclear envelope forms, and eventually the cell enters the cytokinesis, where this protein ring forms, and this protein ring contracts and separates two cells from one cell, forms two cells from one cell. So cytokinesis, the cell splits into two new cells, and the two cells each contain one set of chromosomes. Okay, so it doesn't, these cells do not contain the two pairs of, the two members of a pair in a chromosome. They contain only one. So the cells that result from meiosis 1 are going to be haploid. So meiosis 2 is like mitosis, but with no DNA replication. So between meiosis 1 and meiosis 2, there's going to be no DNA replication. The chromosomes will remain intact. They will just enter into prophase 1, where the nuclear envelope will dissolve. Then they will enter metaphase 2, where these chromosomes will line up along the equator of the cell. And during anaphase 2, the chromosomes are going to be pulled apart by the spindle fibers and be located in the polar opposites of a cell. And during telophase 2 and cytokinesis, the nuclear envelope forms, the protein ring forms, the protein ring contracts, and we have 4 haploid cells. So from one germ cell, there can be created, the four sex cells are created. So each germ cell produces 4 gametes once it undergoes meiosis. So the bottom line is meiosis reduces the number of chromosomes by half. So germ cells have 2 sets of chromosomes, total 4 chromosomes. Let's say if the genotype of this particular cell contains of the 2 pairs of chromosomes, the total number is 4. But after full meiosis, gametes have only 1 set of the 2 chromosomes. So in total, gametes will have 2 chromosomes. If the genotype of this hypothetical organism will consist of the 2 pairs of chromosomes. In human, we have 23 pairs of chromosomes. So our germ cells contain 46 chromosomes, but once the germ cells undergo meiosis, we will produce, it will produce 4 cells with, 4? Yeah. It will have, our gametes have 23 chromosomes. So the third way how genetic diversity is created, with the first way being crossing over, the second way is random assortment. The third way how genetic diversity is produced is random fertilization. So in one mating, any of a mother's possible egg cells can combine with any of her mate's possible sperm cells. So in humans, there can be lots of sperm cells, but there's only 1 egg cell per month. But this egg cell has, more than 8 million of possibilities of what genes are going to be carried in this egg cell. And similar in sperm cells, like more than 8 million possibilities of what kind of genes are going to be carried in the sperm cell. And this random fertilization, this egg being fertilized by a random sperm cell, is another way how genetic diversity is created. So bottom line is there are lots and lots and lots and lots of possibilities of what kind of genetic makeup the offspring will have. All right. So with this kind of genetic diversity, how is it possible that two people can look exactly alike? And we see it pretty often with twins. Okay, and the reason for that, there are two types of twins, and there are identical twins and fraternal twins, right? And they are different. So in identical twins, the two embryos will develop from one zygote. So one sperm will fertilize one egg and forming one zygote. But the zygote, at some point of the development, it will split into two embryos. So instead of one embryo, two embryos will be produced. So the implication of this is in identical twins, the genotype will be exactly the same. So two kids will have exactly same genotype, and it is pretty fascinating. And this property of the twins, of the identical twins, has led to some pretty interesting research. There are lots of so-called twin studies exploring the effect of genetic influence on the person's development versus environmental influence on the person's development. Early research with the twin studies was pretty harsh, but like in the 60s, before the 60s, before the 70s, I would say. Afterwards, it has been much more humane. But so in the early days, lots of parents, not lots of parents, but in the early days, when people gave up twins, the twins would be separated into adoptive homes. Okay, and this separation allowed basically researching how environment influences a person's development. For instance, studies have revealed that the mental health issues are influenced more by the environment rather than genetic makeup. Like when the twins were, there's even the movie was made about it, there were like triplicates that were separated into different homes, and the environment has led to severe mental health issues of one of the member of the triplets. And the other two were completely fine because the environment in which they grew up in was more pleasant, was more supportive compared to the environment in that, unfortunately, of the twin that had severe mental health issues. So also habits are researched thanks to the identical twins, and athletic performance and other talents, etc. So there are some weird ways how the twins are similar, even though they lived apart. For instance, like they have same preferences in clothing or same color preferences. So which points at that these random things like color preferences and clothes should not be genetically influenced, but they are in some way. And it's a mystery why, but apparently these random things are not learned. They are kind of, you can be genetically predisposed to like the color yellow, or you can be genetically predisposed to like a certain pattern in clothing, a certain style in clothing. So the second type of twins are the fraternal twins, and fraternal twins are also called dizygotic twins. And in dizygotic twins, basically there are two sperm cells involved and two egg cells involved. So from two sperm cells and two egg cells, two zygotes will be formed and two embryos will be formed, and there will be just two babies growing in one womb. Okay, so occasionally a woman's organism releases two egg cells and not just one, and if both of the egg cells are fertilized, then the woman will be pregnant with fraternal twins. So the fraternal twins are not genetically identical. They have completely different DNA makeup. It's just so happened that they grew up in the same womb, and that's basically it. So they're basically two different children. It would be the same if the woman got pregnant twice and bore two different babies. All right. Any questions about that? The last topic I wanted to cover for today was non-disjunction of the chromosomes and what happens if there is non-disjunction. All right, so I'm looking through the chat. Feel free to ask your question, unmute yourself and ask a question. So I see the question from Melis. Could be that two sperm enter one egg, right? No, only one sperm cell enters an egg. Two sperms will not enter an egg. I see the question from Unasi. So dogs release multiple eggs, and that's why these have litters of multiple puppies that are not identical. That yes, exactly right. Unasi, dogs release multiple eggs, and that's why they have multiple puppies. So it's basically the puppies are fraternal twins, or triplets, or quadruplets. All right. Can you? A question from Sarah. Can you go over anaphase 1 again, please? Yes. Here we go. Anaphase 1. So anaphase 1, during anaphase 1, the spindle fibers shorten and they pull apart chromosomes in the polar opposite side of a cell. So homologous chromosomes line up along the equator during metaphase 1, and during anaphase 1, the spindle fibers shorten and pull these homologous chromosomes apart into the opposite sides of the cell. But the sister chromatids remain joined. Remember, each X, when the chromosome is depicted as an X, technically it's a replicated chromosome. Chromosome can look like a stick as well, like it's just a single half, like a half of an X. So the X is basically a replicated chromosome. And these replicated chromosomes, one inherited from the mother with, one inherited from the father with a little bit inherited from the mother that was produced by the crossing over, are pulled apart into the polar opposite sides of the cell. Okay, I see a question from Amal. What does homologous mean again? Homologous chromosomes are the chromosomes that contain exactly the same types of genes, but may contain different alleles or versions of the gene. The two chromosomes are homologous if they contain the same type of genes, but these genes were inherited from the mother or the father, but the chromosome itself can come from mother and a father. So why two homologous chromosomes? Why do we have two homologous chromosomes? It's because one was inherited from the mother and the other was inherited from the father. So we have two copies of the same gene, two alleles of the same gene, one that came from the mother and one that came from the father. Okay, quickly remind us the difference between chromatid and chromosome. If the chromosome is replicated and it forms an X, one half of the X is going to be chromatid. So the one replicated chromosome that looks like an X will have two chromatids. One half of the chromosome is a chromatid. Okay. Is the term independent assortment or random assortment? Independent assortment and random fertilization. And that's it. Okay, we'll move on to the non-disjunction real quick. So the meiosis doesn't go always as it's intended to go. Meiosis can go wrong, and what can go wrong is basically the chromosomes may not separate into gametes properly. So they do not separate into gametes properly as a result of non-disjunction. So during anaphase, remember the chromosomes are pulled apart into the polar opposite sides of the cell, but they don't always get pulled apart. Sometimes they're not. So if they're not pulled apart during anaphase 1, then the resulting cell from meiosis 1 can remain diploid, and the other resulting cell during after meiosis 1 can have no chromosomes at all. And as a result, the gametes will have, the half of the gametes will have no chromosomes at all, and the other half will have abnormal number of chromosomes still, like they will still remain diploid. There will be diploid gametes. As a result, if a diploid gamete meets with a haploid gamete, we will have zygotes with three chromosomes instead of 2. Okay, so the zygotes will have 1 extra chromosome if there is so-called non-disjunction in anaphase 1. And non-disjunction is when the chromosomes are not separated in a proper way. Basically, when the chromosomes are pulled to one cell and no chromosomes are pulled into the other cell. And if the empty gamete from one of the parents will meet with non-empty, with a normal gamete in the other parents, the resulting zygote will be missing 1 chromosome. During anaphase 2, there can also be non-disjunction. And as a result of non-disjunction in anaphase 2, there will be still going to be some errors in the zygotes, but there can be a zygote that is missing only 1 chromosome. There can be zygote that is missing that will have 1 extra chromosome, and there can be two normal zygotes. Okay, so if non-disjunction occurs during anaphase 2, there is 50% chance that the zygotes will be fine. But when non-disjunction occurs during anaphase 1, there is no chance that the zygotes resulting from these abnormal gametes are going to be normal. So non-disjunction during anaphase 1 is kind of worse than non-disjunction during anaphase 2, because non-disjunction in anaphase 2 has 50% chance of normal zygotes. So non-disjunction can be lethal. Okay, so extra chromosomes tend to create defects, while missing chromosomes tend to make development impossible. So unfortunately, spontaneous abortions, they are fairly common among women of childbearing age, and pregnancy does not always go smoothly. Okay, so half of spontaneous abortions, half of the pregnancies that are not carried to term, are caused by missing or extra chromosome. So if the zygote does not have a chromosome or has an extra chromosome, basically the body is programmed to get rid of the abnormal cell. Okay, so the embryo or the zygote basically would not develop into a viable embryo, and the body basically kills it. You see, I think it's miscarriage. Yeah, okay, let's call it miscarriage. I think the book calls it spontaneous abortion, but let's call it miscarriage. Okay. Yeah, spontaneous abortion sounds like you spontaneously decided to get an abortion. Poor choice of words. It's weird that the book uses it. And the example of the 1 extra chromosome and the kind of effects that it can have is you can basically evaluate it by looking at by reading about the Down syndrome. It's fairly common. I mean, it's a low chance to have a baby with a Down syndrome, but because there are billions of people on Earth, there are lots of people with Down syndrome. So the Down syndrome is caused by, it's also called trisomy 21. So basically the Down syndrome is caused by an extra chromosome in our 21st pair of chromosomes in the human genome. And it's seemingly not a big deal to have one extra something, but during the development, during synthesis of all the proteins that the cells produce, during synthesis of these vast, during accommodation of these vast amount of processes necessary for survival, somehow this extra chromosome results in in various symptoms. So there's particular appearance that people with Down syndrome have. They have so-called a characteristic line on the palm of the hand, indicating like somehow that is resulting of this extra chromosome in the 21st pair. What else? There is particular like signs in the appearance, and there is mental impairment. It can be mild or it can be severe. There are heart issues, and often, unfortunately, children with Down syndrome die at a very young age. So yeah, this is one example of how having an extra chromosome that was a result of non-disjunction during meiosis can have on a child's development. And so non-disjunction, it happens more, and non-disjunction happens more often as the person ages generally. Like for instance, it's much higher probability that a woman after 40 will have a person with a child with Down syndrome compared to a young woman. And it's not clear, it's not particularly clear to me why, but maybe it's because like the processes in our body do not happen as efficiently as we age. So there's that. All right, it's 10:21. I went a little bit over. If there are any last minute questions, I can answer them. Otherwise, we'll see each other on Friday.
I have a super quick question that's
Yeah.
like just to build off of what you were saying about Down syndrome.
Uh-huh.
There was a study, I believe that it was in Sweden, where they developed a test where they could see if like a baby was going to be born with Down syndrome or not. And I was just wondering if that had like, and if like, I wasn't sure like how they did it exactly. Do you know if that was like through cell research or?
Oh, a long time ago, I participated in like analysis of data that I think that this was kind of data that you're talking about, but I don't remember what kind of tissues they took, whether it was amniotic fluid or what. But yeah, sorry, I don't have a ready answer for that question.
No, it's okay. It was just out of curiosity, but thank you.
Yeah, no problem. All right,
Oh.
guys, go ahead.
I'm gonna
Actually,
say I looked up what you were talking about, and it looks like a lot of times that they just look in an ultrasound when the baby is developed enough to tell. But they did mention that they're trying to advance genetics to be able to test for that kind of thing. So
there is some sort of test. I mean, it's pretty common in Europe. I mean, I worked on it. I worked on data in Ukraine, and like literally every pregnant woman took this test, and it was certain probability. Like this test could tell with a certain probability whether the baby would have genetic defects, including the Down syndrome. But yeah, I'm just blanking on it. I'll get back to you, Dylan, on that.
Okay, thank you very much, both of you.
All right,
Real
guys.
quick, I don't know
Yeah,
the name
go
of
ahead,
it.
Brandy.
Sorry, I don't know the name of it, but in all of my pregnancies, I've had to do like blood work to get done, and in one of the options, there's an option to see if there's a possibility of like Down syndrome through the blood work.
Do you remember what it was called?
I can look it up and let you guys know on Friday.
Ooh, please do. Yeah, person with
Okay.
experience.
Thank you.
Yeah, no problem. Let you know.
Cool. Thanks, Brandy. All right. Thanks, guys, and I'll see ya on Friday.